Hello friends, this is the second video of immune response. Let's begin with cell-mediated immune response. The term cell-mediated immunity refers to specific required immune responses mediated by sensitized T cells. This form of immunity can be transferred from donor to recipient with intact lymphocytes but not with the antisera, hence it is called cell-mediated immune reaction. Cell-mediated immunity, that is in short CMI, plays an important role in some immunological functions. Let's know about these immunological functions. First is delayed hypersensitivity, that is type 4 hypersensitivity, immunity in infectious diseases caused by intracellular organisms, transplantation immunity and graft versus host reaction, immunological surveillance, and immunity against cancer. Pathogenesis of certain autoimmune diseases, example thyroiditis. Now let's know about induction of CMI. Foreign antigen is presented by antigen presenting cells to T lymphocytes. T lymphocytes possess antigen recognition receptors known as T cell receptors that recognize foreign antigen and a self MHC molecule on the surface of the APC. These sensitized T lymphocytes undergo blast transformation, clonal proliferation, and differentiation into memory cells and effector cells, that is THTC, TDTS. The activated lymphocytes release biologically active products, that is lymphokines, which are responsible for various manifestations of CMI. T cell T cells recognize antigens only when presented with MSC molecules. CD8 plus cells can recognize the combination of foreign antigen and class 1 MSC antigen and differentiate into TC and TS lymphocytes, whereas CD4 plus cells can recognize the combination of foreign antigen with class 2 MSC antigen and differentiate into TH and TD cells. TC lymphocytes recognize foreign antigen and class 1 MSC antigen and gets attached to the target cells. This stimulates the TC lymphocytes to release cytolysis lysins, which leads to lysis of the target cell. Subsequently, the TC cell may detach from the target cell and repeat the same process with another. TC lymphocytes also synthesize and secrete interferon gamma, and thus they probably also contribute to some extent of macrophage activation. Now let's know about cytokines. These are biologically active substances secreted by monocytes, lymphocytes and other cells. They are named lymphokines if they are derived from lymphocytes and monokines if they are derived from monocytes and macrophages. Interleukins are chemical substances that function primarily as growth and differentiating factors. They exert a regulatory influence on other cells. All these biologically active substances, that is lymphokines, monokines and interleukins, and they are not specific for antigens. Some of the important cytokines we will now discuss. First is interleukin 1. Interleukin 1, in short IL-1, is principally secreted by macrophages and monocytes. It occurs in two forms, that is IL-1 alpha and beta. Its production is stimulated by antigens, toxins, inflammatory processes and inhibited by corticosteroids and cyclosporin A. It is a stable polypeptide. Now let's know about immunological effect of IL-1. Stimulation of T cells for the production of IL-2 and other lymphokines. B. Cell proliferation and antibody synthesis. Third point is neutrophil chemotaxis and phagocytosis. Together with the tumor necrosis factor, in short TNF, it is responsible for many of the hematological changes in septic shock. Second point is interleukin 2. Interleukin 2 is the major activator of T and B cells and stimulates cytotoxic cells, in short TC cells and NK cells. It converts certain null cells, that is LGL, into lymphokine activated killer cells, in short LAK cells, which help to destroy NK resistant tumor cells. This property has been used in the treatment of cancers. IL-2 was previously named as T cell growth factor. It is produced by T cells. Interleukin 3. Interleukin 3 is a growth factor for bone marrow stem cells. It is also known as multi-colony stimulating factor. Now let's know about interleukin 4. Interleukin 4 acts as B cell differentiating factor. It activates resulting B cells. It also acts as growth factor for T cells and mast cells. It augments IgE synthesis and therefore may have a role in atopic hypersensitivity. It enhances the activity of cytotoxic T cells. It was formerly known as B cell growth factor 1. Interleukin 5. Now let's know about interleukin 5. Insert IL-5 causes proliferation to activate B cells. It was formerly known as B cell growth factor 2. Now let's know about interleukin 6. Interleukin 6 is produced by stimulated T and B cells. It is also produced by macrophages and fibroblasts. It induces immunoglobulin synthesis by B cells and formation of IL-2 receptors on T cells. Now let's know about colony stimulating factors. In short, CSF. They stimulate the growth and differentiation of pluripotent stem cells in bone marrow. They are named after the types of cells they induce. For example, granulocyte, that is for G, or mononuclear, that is MCSF. IL-3 induces growth of all types of hematopoietic cells and thus known as multi-CSF. They are responsible for adjusting the rate of production of blood cells according to requirements. Example, massive granulocyte response is seen in pyogenic infections. Now let's know about tumor necrosis factor. In short, TNF. The tumor necrosis factor occurs as two types, namely alpha and beta. TNF alpha is produced principally by activated macrophages and monocytes. It resembles IL-1 in having a very wide spectrum of biological activities, such as its role in the manifestations of endotoxic shock. It also has immunomodulatory influences on other cytokines. TNF beta is formed principally by T helper cells. Its actions are similar to those of TNF alpha. TNF beta was formerly known as lymphotoxic interference. Now let's know about interference, in short IFN. There are three classes of IFNs: alpha, beta and gamma. IFN alpha is produced by leukocytes, beta produced by fibroblasts and gamma is by activated T cells. IFN gamma increases the cytotoxicity of NK cells and macrophages. It has inhibitory effect on malignant cells. Interferons also have antiviral activity. Cytokine production is regulated by exogenous factors such as antigens as well as by endogenous factors. They also regulate each other by positive and negative feedbacks. IL-1, 2, 3, CSF and IFNs have already found therapeutic application. Now let's know about lymphokines. Migration inhibiting factor. It inhibits the migration of normal macrophages. Macrophage activating factor. It restricts macrophage movement and increases phagocytic activity. Now let's know about macrophage chemotactic factor. It stimulates chemotaxis of macrophages. Macrophage stimulating factor. It stimulates macrophage migration to the site of action. Now let's know about detection of CMI. CMI can be detected by some methods. First is skin test for delayed hypersensitivity. Second is lymphocyte transformation test. Third is migration inhibiting factor test. Fourth is rosette formation. Fifth is detection of T cells by immunofluorescence technique. Now let's know about skin test for delayed hypersensitivity. It is useful to detect delayed hypersensitivity to commonly encountered antigens. Tuberculin skin test is commonly used. Other antigen used for skin testing are 1% solution of 1-chloro-2,4-dinitrochlorobenzene or dinitrofluorobenzene, in short DNFB. Second is lymphocyte transformation test. This is in vitro test which detects transformation of cultured sensitized T lymphocytes on contact with specific antigen. Now let's know about migration inhibiting factor test. Human peripheral leukocytes are incubated in capillary tubes in culture chambers containing culture fluid. In the absence of antigen, the leukocytes migrate out to the open end of the tube to form a fan-like pattern. When an antigen to which the individual has CMI is introduced into the culture fluid, the leukocytes are prevented from migrating. By comparison with the control, it is possible to make a semi-quantitative assessment of the migration inhibition. Now let's know about rosette formation. Most T cells form rosettes when incubated with sheep erythrocytes. Rosette is a lymphocyte to which 3 or more sheep erythrocytes are attached. T cell rosette is called as E rosette. T cells can be estimated by counting E rosettes and thus help in detection of CMI status. Now let's know about detection of T cells by immunofluorescence technique. T cells can be detected by immunofluorescence technique using monoclonal antibodies. Now let's know about transfer factor. Lawrence reported transfer of CMI in man by injecting the extract from the leukocytes. The extract contains a soluble factor known as transfer factor. The transferred immunity is specific as CMI can be transferred only to those antigens to which the donor is sensitive. TF is a low molecular weight substance. That means molecular weight will be 2000 to 4000, resistant to trypsin, but gets inactivated at 56 degrees centigrade in 30 minutes. It remains stable for several years at 20 degrees centigrade and in the lyophilized form at 4 degrees centigrade. It is not antigenic. Chemically, it appears to be polypeptide polynucleotide. It is highly potent. The transferred CMI is systemic and not local. Humoral immunity is not transferred by transfer factor. The mechanism of action of TF is not known. It probably stimulates the release of lymphokines from sensitized T lymphocytes. TF has been used in T cell deficiency, that is Wiskott-Aldrich syndrome patient, treatment of disseminated infections associated with deficient CMI, and third is treatment of malignant melanoma and other types of cancer. Now let's know about immunological tolerance. Immunological tolerance is defined as a state in which contact with an antigen specifically abolizes the capacity to mount an immune response against that particular antigen when it is administered subsequently, the immune reactivity to other antigens being unaffected. Immune tolerance may be of two types, natural or acquired. Natural tolerance is non-responsiveness to self-antigens. Any antigen that comes in contact with the immunological system during its embryonic life would be recognized as self-antigen and would not provoke an immune response in the mature animal. Bonnet and Fenner in 1949 postulated that tolerance could also be induced against foreign antigens if they were administered during embryonic life. And acquired tolerance arises when a potential antigen induces a state of unresponsiveness to itself. For acquired tolerance to be maintained, the antigen must persist or be repeatedly administered. The induction of tolerance depends on the species and immunocompetence of the host. Physical nature, dose, and route of administration of antigens. Rabbits and mice can be rendered tolerant more rapidly than guinea pigs and chickens. It is difficult to induce tolerance when the degree of immunocompetence of the host is higher. Soluble antigen and heptins are more tolerogenic than particulate antigens. For example, when human gamma globulin is heat aggregated, it is immunogenic in mice, but when deaggregated, it is tolerogenic. Now let's know about finally finished. Any questions regarding this, you can ask me in the comment box and subscribe my channel to know more updates. Thank you.